All right, so this is lecture 11, and in the last class we had completed our short section on nucleic acids. And we looked at the structure of double-stranded DNA, and the structure of double-stranded DNA is very interesting. There's this phosphodiester backbone, which carries a strong negative charge. And then where we see the orange are in these two kind of grooves. The big groove is the major groove, and the smaller groove is the minor groove. And we care about these grooves because if we look inside them, you can actually see the bases. And what that means is this is a place where proteins or other molecules can interact specifically with the bases. And remember, one of the theories was that the structure of DNA would be such that the bases were sticking out. And it turns out they're inside, more like a ladder, but there's still access. bDNA is the most common form of DNA. It's not the only structure, but it's really the only one we're going to concentrate in this class. We have two anti-parallel strands. So we talked a little bit about the polarity of the DNA strands, and we know that there's a 5' and a 3' end. They're all arranged in the same way, so 5', 3', 5', 3', 5', 3', and then the molecule is made in a 5' to 3' direction. One of the major forces holding these together and the specificity between the base pairing. So that specificity is determined by the hydrogen bonding, where we have 3 hydrogen bonds between G's and C's and 2 between A's and T's. The second strand of the DNA runs in an anti-parallel direction, so 5', 3', here's the 5' end, here is the 3' end. Okay. So that's the major structure of the DNA. We can call this base pairing Watson-Crick, and you see that used commonly, but it's more correct, I think, to call this canonical. Canonical meaning that it follows the rules, or it's the most common. And so here we can see the differences between the GC and the ATs. What we notice in both cases is that we have a purine. Okay, so this is a purine with a pyrimidine, and then same here. And the difference is 3 hydrogen bonds versus 2. And so actually sections where it's GC rich in the DNA, the DNA strands are held together a little bit more tightly. And we talked about how this base pairing then also allows for semi-conservative replication, which was a concept we talked about in, I think, our second or so lecture. We move from there to talk about carbohydrates, which we'll continue on today. So carbohydrates, really, it's the structure that is, so the structure of different carbohydrates and the way they put together, there's a huge variety there, much more than we've seen with nucleotides or proteins. And there's different ways that we classify or name them. One of the ways we name them is often based on the number of carbons. Okay? Keeping in mind that the molecular formula here is CH2ON, so you have a water for every carbon, which is the name, hydrates of carbon. And then the next important one we talked about was the location of the carbonyl. If it was on the end, if it's on carbon 1, that makes this an aldehyde chemical group. We call these aldoses. Otherwise, it'll be on carbon 2 to make a ketose, because this is a ketone here. And that was basically where we got to in the last class. Today we'll be finishing our section on carbohydrates, and we'll be looking at how they're put together. Again, we're kind of trying to keep it brief through here because many of you will cover this in more detail in biochemistry courses, Biochem 300A or 299. But we need to know the importance of it a little bit to understand some of the concepts we'll be dealing with a little later in the course. We'll then look at fatty acids and membrane lipids. So your exams, I have to make sure students that miss the exam have until the end of the day today to let me know about it. And once we've got that all figured out, I'll get your marks to you. If no one's writing the deferred, you'll get the marks this week. Otherwise, you'll get them either on the weekend or Monday. So again, we'll see how everyone did on it. So don't forget, tomorrow we have our first social hour, so I highly encourage people to come. It's a great opportunity to interact in an informal, casual setting with different scientists. This case, we have Yvonne Stevens. She works for an immunotherapy company in Seattle. So many people know a little bit about immunotherapy, but she's going to give a description of it. And she's also going to talk a little bit about her pathway, how she ended up in Seattle doing what she's doing. And so she's a very dynamic speaker, and I think you'll find it a great story. So the link for that is just the office hours lecture link that we have on Brightspace. Again, you're encouraged to participate in the discussion, and everyone is welcome. So are there any questions from the last stuff? Seems like a long time ago, doesn't it? Way back when. Okay. So the way we classify the structure of carbohydrates is, again, our good old friend Fisher projections. And so at the bottom here, again, this is glyceraldehyde. This is the structure, remember, that we use, glyceraldehyde. We use that to determine the absolute configuration of our amino acids. So here we have the D and the L conformation. The top is like a more proper, this is the way we're used to seeing structures. And when you have a solid line, okay, or not like a solid like these, what does that mean? Yeah, it's coming right at you. Oh my gosh. It's coming right for me. And then what are the dotted ones? They're being pushed away, right? So we can visualize this molecule, hopefully with practice, and we imagine what this can look like in three-dimensional space. Okay? And that's one way we can draw them at the top. The Fisher projections at the bottom are actually a little bit different, okay? So here's the key to a Fisher projection. When we look at a carbon, any carbon, vertical lines are always pushed away. Always pushed, in this case, into the board, the wall. Horizontal lines for a Fisher projection are always coming out. And that might seem weird right now, but we'll figure out why this is in a second. Okay? And so here we have trioses, 3 carbons. The left we have 2 aldoses, so they have the D and the L conformation. Okay? On the right, though, we have dihydroxyacetone, which has no chiral center. So the ketoses have 1 less chiral carbon. So if you're looking at comparing 2 sugars and aldose and a ketose that have the same number of carbons, the ketose will always have 1 less chiral carbon. Just again, different ways that this can be represented. And the center here, this is the notation that you'll generally see used for carbohydrates. All right? And so the chiral center that determines if it's D or L is the chiral center that is, I'll do it on this one, farthest from the carbonyl carbon. So here's our carbonyl carbon. This is a chiral center, okay? You can visualize that. It's got 4 different groups. 1, 2, 3, 4. Those are all different. If we were instead to compare to this carbon, it's not a chiral center because it's bound to 2 hydrogens. So the one that's farthest away is this one, and that determines if it's D or L. And the way we draw this is if our hydroxyl group on that carbon is to the right, we call that the D configuration. And if it's on the left, we call that the L configuration. And this is a standard notation. So this is how you'll always see them drawn. Okay? If you're looking at a single carbohydrate, just one carbohydrate, they'll always be drawn that way. And there, that's all we need to know for this course. Okay? So you'll always be able to recognize then the conformation for this. So if we're looking at this one on the left, okay, who wants to tell me the number of the chiral carbon that's going to determine if it's D or L? What number carbon? 1, 2, 3, 4, 5, or 6? Yes? It's not 2. No, it's the farthest away. So it is... Can't be 6, because 6 isn't chiral. 5, right. So this is the farthest away chiral carbon in this example. So, is this D or L? Who thinks this is in the D conformation? And you're all correct. Everybody picked D. Good work. So this is a D sugar. Most sugars are in the D conformation. It's not as strict as we see with amino acids, but I think all of the examples that we look at in the class are D sugars. So I'm gonna sort of sweep a few things kind of under the rug and explain the structure. I don't want to spend a lot of time on it because I think this isn't really the place for it. We'll cover it in biochemistry classes, and you've maybe seen it in chem courses before. But we will spend a couple of minutes on it because there's a few important things we have to get out of this. The first thing is that this structure, as we see it, remember all these vertical lines are all pushing into the board. Okay? That's not how we usually draw a structure. And so if we follow this around, what this Fisher projection is doing, can we imagine, it's a whole bunch of kinked turns. And what shape am I forming as I do that? It's actually forming, that linear molecule is actually bending around, almost like an open ring. Okay? I used to have molecules to do this, but as I said, I lost them all during the, somewhere in my house or office during the COVID boom. This all got lost. So because each of those vertical lines is pushing into the board from that carbon, what it ultimately means is here's a carbon, and now I'm taking it from this, and I'm turning it on its side so we can see what's happening. So here's a carbon, pushed in, pushed in. This carbon, pushed, rotate, pushed in, and so we end up forming almost a ring. And the reason that's important is when we're looking at carbohydrates, they have 4 or more carbons, they're most likely to be in a ring form. And the carbons we're really interested in, or the carbohydrates we're often interested in, very often have 5 or 6 carbons. And so the reality is that they're almost always in a ring formation. Okay? And so this notation, the Fisher projection, lets us predict what that ring looks like. And the prediction that we can make from that is, well, first let me tell you how the ring forms. The ring forms because a hydroxyl group, OH, can attack the carbonyl carbon. And this will then go off and pick up a hydrogen to become an OH group. Okay? So our carbonyl carbon, which is carbon 1 for an aldose, 2 for a ketose, is going to end up with a hydroxyl group that's stuck to it. And one of these hydroxyl groups is the one that's going to attack it. And commonly, so we're looking at glucose here, the hydroxyl group on position 5 is the one that attacks. So this oxygen here, oops, wow, that was exciting. This one here is going to be a link between, let me change the color here, if I can. Okay, so far people are okay with that, right? So that hydroxyl group will actually come around and attack the carbonyl carbon, forming a covalent bond between, so it'll form a covalent bond between this oxygen and this carbon. Okay? So keep your eye on the numbers. So here was carbon 5. Here's the oxygen that was on carbon 5, and it's formed a covalent bond with that carbonyl carbon at position 1 for an aldose or position 2 for a ketose. Okay? And so this then, the bridging oxygen, actually comes from one of these side chains. And again, we're mostly dealing with 5 or 6 carbon sugars. And with those, usually it's carbon 5 or carbon 6, the hydroxyl group that will attack, because it's more, there's less steric hindrance. So let me erase that again. So far so good, right? So we remember that carbon 1 or 2, the carbonyl carbon, is always the one that gets attacked. So it's always that carbon that's attacked, the carbonyl carbon. Right? And it can be attacked by potentially any of the hydroxyl groups, but only certain ones are sterically allowed, because otherwise you have to bend the ring too much. Okay? Yes?
I'm sorry, I just wanted to clarify. It's the oxygen that covalently bonds with the carbon.
Always the oxygen, which is why when the ring closes, there's this, we call this an ether linkage. And an ether is where you have this. So we end up with an ether linkage between the attacking carbon and the carbonyl carbon. So when this rearranges, this has to go looking for a hydrogen, which it's going to pull off of water. Okay? But what that means is that when it picks it up, this new group, okay, which is going to become an OH in our new molecule, will either end up below the ring or above the ring. So what do I mean by above or below the ring? Well, when we take our molecule, and here's why we're going to learn to love, Fisher projections. If we take our molecule and we count around the carbons on the ring, the way our Fisher projection works is everything to the right, okay, ends up below the plane of the ring. So carbon 2, below the plane of the ring. Carbon 3 is on the left, so that's above. Carbon 4 to the right, below. Carbon 5, if it's in the D conformation, what we have is the little tail comes up, the CH2OH, and is above the plane of the ring. Okay? So if it's in the D conformation, which this one is, this will be above the plane of the ring. If it's in the L conformation, it'll be below the plane of the ring. So what we can do is, looking at the Fisher projection, we can see the location of the hydroxyl groups. Where did this hydroxyl group go? This OH. Where's that one? Yeah, perfect. That's this one. Right here. Good. Okay? So this, though, was a carbonyl, and it picks up a hydroxyl group. And that means that that one, depending on which way it breaks, either ends up below the plane of the ring or above. And to keep it easy, if it's below, they call it alpha, and if it's above, they call it beta, which is the total opposite of what would make sense. And so we call this carbon, and I do have a slide coming up for this, but I don't want to wait because it's so exciting. We call this carbon that used to have the carbonyl, used to be the carbonyl carbon. Okay, this was the one that determined if it was an aldose or ketose. We call this, when it's in the ring form, we call this the anomeric carbon. And we'll see why we care so much about this anomeric carbon in a few minutes. But this is something we need to understand. So we get this attack. Then our oxygen goes looking for a hydrogen. When it gets it, it either breaks above or below the plane of the ring. All right, if it's above, we call it beta. Below, we call it alpha. We have a couple of ways that we can draw this. We can draw it how it structurally looks, which is not flat, but instead, we utilize this method of drawing often called Hayworth projections. So the way these work is the thick part of the ring is pointing towards you, and the thin part is going off into the background. And I think most people, just from looking at this before me even describing that to you, could probably make out that that's what was happening in here. This isn't the actual shape of the carbohydrates. The ring is not planar, meaning flat. It actually is bent. And there's different ways that it can bend and twist to fit in, sterically, to fit in all of the molecules. So, I don't think we'll use this slide. This was just to show, what happens if we have a ketose. We can have the same attack going, but I don't think we'll worry about this one this year. So we've got enough on our plates. Yes?
Sorry, so the attacking hydroxide, where does the hydrogen on that one go?
That one's just lost during the reaction. Yeah. So this is describing the two different anomers. The term pyranose here, so we're starting with glucose, is the linear molecule. The term pyranose, this is a pyranose ring. Okay? And a pyranose ring has 1, 2, 3, 4, 5, 6 members in a pyranose ring, one of which is that oxygen. So the pyranose has 6 components in the ring. When we're looking at something like ribose in DNA, it's got 5 members in that ring, and so that's called the furanose, F-U-R-A-N-O-S-E. So these two anomers then are the two types of glucopyranose. And keep in mind, they're structurally distinct. So that location of the hydroxyl group, the anomer, they're structurally different, which means there's differences in how they're recognized and everything. Okay? Yes?
Sorry, the word kernels, that just means how many members it has, right?
Yeah, that just tells us how many members in the ring. So that tells us that's a cyclized glucose, and there's 6 members in the ring. Okay, we're going to step back from this for a minute, and then we're going to come back and talk about the importance of that anomeric carbon and why I spend so much time on it and why we care about it. So there's a whole bunch of written description here, but I think it's better to just kind of hop ahead to this terminology visualized chart. And this describes the different structures that we can have for carbohydrates. So if we look at the term isomer, that just means that 2 molecules have the same molecular formula. So they have the same number of carbons, same number of hydrogens, same number of oxygens. Constitutional isomers are not connected in the same order. So if we're looking at carbohydrates, for example, here we have 2 trioses, glyceraldehyde and dihydroxyacetone. They have the exact same chemical makeup, but for the glyceraldehyde, carbon 1 is the carbonyl carbon, and for dihydroxyacetone, it's carbon 2. So they're put together in a different way. When we move to stereoisomers, the atoms are connected in the same order. So for example, here in the enantiomer section, carbon 1 is a carbonyl carbon. Carbon 2 has a hydroxyl group. Carbon 3 has an alcohol and 2 hydrogens. So they're connected in the same way. If they're connected in the same way and they're mirror images, we call those enantiomers. So these would be the D and the L isomers of a given sugar. So the D and the L just look like mirror images. So if we dropped a mirror down the center, they would each be each other's reflection. We're not really going to see any examples of that in this course. What we will see is diastereomers. Diastereomers, basically where the hydroxyl groups are differ, whether they're to the right or the left of the molecule. Okay? And so there's a problem in this figure that we should correct, and that should be a H there and not an OH. Because actually, if we were to look at this, these aren't even isomers because they actually have a different molecular formula. So it's just a printing error from the textbook. So this should be an H. So what we can see is carbon 1 is the carbonyl carbon in both. 2 has a hydroxyl, 3 has a hydroxyl, and so on. So they are stereoisomers. But they're different in that here we have the hydroxyl group to the left, so that's going to be above the plane of the ring. Below, below. Here we're below, above, below. And remember, 2 things that have different structures have different functions. We're not going to worry about epimers, so you can just X that out. Again, really, we're not going to see enantiomers. And then here we have the anomers. So the anomers are identical except for the location of the hydroxyl group. And that anomeric carbon, okay, the anomeric carbon in an unmodified carbohydrate, which will be the examples we'll see, will have a hydroxyl group on it. Okay, so why do we care so much about the anomeric carbon? Well, we can link that OH, that group, that carbon that's attached to the hydroxyl, we can link to we can link that to a hydroxyl group or an amine on another molecule. And what's something that has a lot of hydroxyl groups? Carbohydrates have lots of hydroxyl groups. So through the anomeric carbon, we can link carbohydrates together via the hydroxyl groups. On this slide, what's really important to note, and this is a very important take-home, when you are attaching carbohydrates to other carbohydrates, to amino acids, or to lipids, those are the three groups we're going to talk about, the linkage for one of the carbohydrates at least always has to be through the anomeric carbon. So this is why I stressed and spent so much time on anomeric carbons. For one of those carbohydrates, the link always has to be through the anomeric carbon. And so what we can see for sucrose, these are disaccharides, meaning there's two of them, and there's lots of disaccharides that are quite important. Sucrose, so this is table sugar. What I can see here is this is the anomeric carbon on glucose. And actually, this is the anomeric carbon on fructose as well. Okay? But let's look here. Here, we have the anomeric carbon of the left-hand glucose, not to the anomeric carbon, but to carbon 4 of another neighboring glucose. This is important because this glucose molecule has another anomeric carbon. So it can potentially be linked to another carbohydrate or an amine group. Okay? And so if we look at all of these, we can see one of the linkages is always through the anomeric carbon. If a sugar has a free anomeric carbon, free hydroxyl on the anomeric carbon, it can potentially be linked to something else. And it turns out that they're more stable when they're bound through their anomeric carbons to something else, for reasons we don't need to get into. Okay? Questions about that? It's tricky, but I think at this sort of level of detail, hopefully people can understand how we get the anomeric carbon, the difference in the alpha and the beta, and then importantly, the fact that the anomeric carbon can be used to link to other molecules. And that's a covalent linkage, so a stable linkage. Yes? The 4 on glucose is not the anomeric carbon, no. And actually, we can go through and look at that. Okay. So this, carbon 1, 2, 3, this is carbon 4. The anomeric carbon will always be carbon 1 for an aldose and carbon 2 for a ketose. And if I'm ever asking you to do these sorts of things on a quiz, I will always number the carbons for you, so you know which carbons are which. So there, free marks. Carbon 1 if it's an aldose, carbon 2 if it's a ketose. So oligosaccharides means that there are a small number. Oligo means small number. We'll look at a couple of examples of oligosaccharides when we're talking about something called the endoplasmic reticulum. And polysaccharides are large sugar molecules. So what's interesting about polysaccharides compared to oligosaccharides, the oligosaccharides we'll look at will often have a bunch of different types of carbohydrates, And they'll be linked through different carbon combinations. When we look at polysaccharides, they're often sort of dull molecules because they'll have one or two subunits making them up, and the linkages will be repeated over and over again. So what do I mean by that? So if we take glucose, glucose, of course, is a fuel molecule. And it can be produced by plants. All right, we can consume it, but us and plants can also store it. And the thing about storing solutes in an organism is that it sets up a concentration gradient, and if you have too much solute in a cell, water floods in and bursts the cell. So the way a cell gets around this, okay, so we remember, I don't think we discussed that in this class, but I think you remember that from physiology or other courses where you do that. So if you have a cell and you have a really high concentration of sugar in that cell, what happens? Water moves into that cell and it bursts. Who remembers that? Everyone, right. Okay, mostly everyone. Okay, so if we had that same number of sugar molecules, 10,000 glucose molecules, this is the super weird thing about how osmosis works. If we put those together and build a single starch molecule in a plant, it's recognized as a single molecule. So now there's no longer a concentration gradient, which is kind of hard to wrap your head around because it's the same things, but from an osmotic point of view, it counts as one molecule. So we store glucose in these huge molecules. These huge molecules in plants we call starch, and there's two types. The first is amylose, and we have what's called an alpha-1,4 linkage. So here is the anomeric carbon, carbon 1. It's an alpha anomer because it's below, and it's linked to carbon 4 of the next sugar. This next one, same thing. Here's the anomeric carbon, 1. It's alpha, and it'll be linked to carbon 4 of the next glucose. And this will continue until it makes a huge chain. So this is what I'm saying. It's made up of a single thing, glucose, and it's all the same linkage, alpha-1,4, 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 alpha-1,4. So in polysaccharides, this is what we tend to see. It's not like DNA, where DNA we're using a template, remember? This is not templated. Okay, so the size of it can vary. There's different determinants of size, but it's not reading a template as we saw with DNA, RNA, or protein. So we can call that non-templated. Amylose is often in the form of amylopectin, where about every 30 residues, every 30 sugars, we have an alpha-1,6 linkage. Okay? So here's our anomeric carbon, so carbon 1, and that's through carbon 6 of the other molecules. So this is an alpha-1,6 linkage. Again, that's repeated, so it's a little bit on the dull side. So that's starch. People's starch is something called glycogen. It's the same, it's all alpha-1,4 linkages, but about every 10 or so, we have an alpha-1,6. So it's a little bit more branched. Yes?
Sorry, I'm just wondering if you could repeat what we were saying about templated molecules.
So if we look at DNA, and we're copying DNA, it's done through base pairing. So if we have a stretch of DNA that's 100 bases, the newly made one's 100 bases. If we were to transcribe that into RNA, it would be 100 nucleotides of RNA because you're using it as a template, and you're directly using it as a template. Remember, we pull that DNA apart, and we physically bind the bases to it to make it. And then we're physically doing that to make RNA, and then physically using it as a template to make protein. This isn't templated. It's just driven by enzymatic reaction. So, yeah. And so people's starch is glycogen. And again, it's alpha-1,4s with beta-1,6s. Cellulose. So this is like dietary fiber. What do we know about cellulose compared to starch? Can we eat starch and break it down? No problem. Eat a big bag of starch. Feel good all day. Cellulose goes right through you. Like some bacteria will break it down, but we can't break it down. Exactly the same, 1,4 linkages, but they're beta 1,4 linkages. So exact same molecule, glucose, but it's in the beta conformation and they're linked beta 1,4. And this then gives trees the strength to form trees. And you can chew away and you won't be able to break those down. So that's the importance of that. Often we'll see modifications to carbohydrates, so they won't fit perfectly into our CH2ON. Here's a couple here where we see where we would normally expect a hydroxyl group here. We have what's called an N-acetyl group. And we're linking these through beta 1,4s, and we make a very special polysaccharide called chitin. And this, for example, gives crabs that super hard shell, or other shellfish. Often have a chitin shell around them. So made up of sugars. They don't dissolve in the water. You can't digest them. And here we have the beta 1,4 linkage and also a further modification. And we'll see other examples where we have modified carbohydrates. We're going to talk about lipids in the next class, but we can add carbohydrate groups to lipids. Okay? And so those are generally going to be added through an alcohol, an OH group on the lipid. Will interact with the anomeric carbon. It has to be through an anomeric carbon. And we're also going to see later in the class glycoproteins. And for these glycoproteins, we are going to attach via the anomeric carbon, okay, we're going to attach these sugars to serines or asparaginins. Okay? And we'll see how that works a little bit later. Okay? All right. So is there any questions about that? I'll introduce the topic of lipids and then we talk about the importance. So if we're looking at a lipid is a molecule that is generally more soluble in a hydrophobic solvent than a polar solvent. So you can dissolve more of this stuff in a hydrophobic solvent than a polar solvent. And that's just the simple definition of a lipid. There's lots of different types of molecules that can fall into this. We'll be looking at fatty acids, as well as the glycerides, monoditriglycerides. Importantly, when we're looking at a cell biology course, many of the membrane lipids, okay, membranes are composed of lipids, often phospholipids, glycolipids, but also sterols. Steroids can be important signaling molecules, and then waxes we're not really going to talk about. They're excellent fuel molecules, okay, but they're also an excellent way to store fuel. So we store a lot of energy as fat. And the reason we do that is it's a lot more efficient to store energy as fat than as carbohydrates. So then instead of storing it as glycogen, people starch, we tend to store it as fats because they're more efficient. They're smaller in size and they have more energy per gram. They're important for membrane formation, communication. We'll talk a little bit about steroid signaling later. Things we won't really talk about are physiological or physical, so protection of organs, thermal insulation. Those are also important features of fats. And we'll look at the structures of these in the next lecture. So hopefully, well, we'll see everyone tomorrow, and hopefully we'll see many of you at our scientist talk.